Okay, so welcome everyone, our friends from Pakistan. Good morning from India and from the areas where this morning. Good morning to everyone. For us, it's like for me it's 10:30. It's almost the same for Bob and for other colleagues. It will be slightly later. So we welcome you to this seminar series. Today, we'll be having a talk on ecological and physiological considerations related to C3 and C4, photosynthesis in Neogene ecosystems. Thure Cerling, who's from University of Utah, Department of Geology, he'll be talking about this topic, and it's probably Bob can correct me. I'm not sure if this is 17th talk in this series, and
Yes.
we had been able to put the links to the previous talks for our colleagues or friends who want to retrieve them, so they can go back. And I must have, or I might have provided the link for a previous talk, and we'll be uploading this talk as well. Bob will discuss about the future talks, and he'll introduce the speaker for tonight. Bob?
Yeah, thank you, Irfan, and thank you all for joining us this evening or this morning, wherever you are. And I just want to give a little bit of a look into the future. Next week, we've got Shahid Iqbal from the Quaid-i-Azam University, and he's going to be talking about the provenance of sediments in the Peshawar Basin. And then Yanni Naiman will talk the following week, and she'll probably talk about sort of the regional picture of the Siwaliks between Pakistan and India. And then Advait Junkar will talk on the following, and I think he'll be talking about more biostratigraphy of the Siwaliks. And then we're anticipating having perhaps a final lecture for this series, which would be about the 14th of July. And we're hoping that Dr. Imran Khan from the Geological Survey of Pakistan will be joining us for that final session. And then we'll probably have a break for the summertime. And we're not quite sure whether we will continue in the fall. It'll be a little bit up to the feedback we get from participants. So that's sort of charting out the future. And today, of course, it's a tremendous pleasure for me to welcome Thure Cerling to our seminar. And I first met Thure in, I think it was probably 1972. I was an undergraduate student at the time, and we were working in Lake Turkana, on the east side of Lake Turkana. And Thure was probably a very dignified graduate student. And Thure and a couple of his colleagues were sort of mentors to me, or role models. And I've followed Thure's career, of course, over the last many years. And it's been a wonderful inspiration to see how subtle geochemical information can be teased out of the rocks and out of the fossils to be used to interpret paleoecologies and paleoenvironments. And without any further amplification, I'll just turn it over to Thure with the idea that we'll have about a 40 or 50-minute seminar, and then please feel free to ask any questions at the end, and we can stay a little bit if people want to have an ongoing discussion. So we'll turn it over to Thure.
Okay, thanks. Thanks, Bob. I didn't really know what sort of all the sorts of things that I was going to cover in this talk today. So this talk has been evolving really until about an hour ago, and in fact, after I got on the talk, got on the video about an hour ago talking to Bob, he suggested, oh, add some more slides. So this is probably the latest of any talk I've ever given to have a revision made. The final revision was made only minutes before now. I'd like to give a couple of particular acknowledgements. The Geological Survey of Pakistan, of course, has been incredibly important in this. They supported the field work that was done by the Harvard group, and I'm just mentioning the general Harvard group there. Those two groups just were so good at providing the facilities for this work, and in particular at Harvard, the Harvard group included, in particular, John Barry and Dave Pilbeam, Larry Flynn, and Catherine Badgley. And that group really welcomed me and a graduate student when we first came out in the mid-1980s. Yang Wang was a new graduate student of mine who plays a minor role in this talk, but I did want to mention her. She's a faculty member at Florida State University and never worked in Pakistan, but did some key work understanding the diagenesis that allows us to use fossils in the way that I measure things with stable isotopes. Jim Elringer has been a fabulous colleague of mine. He's a botanist, and I'm a geologist, and we are at the same university. And he's just been a really good colleague in answering my questions about how biology works. Jay Quaid was a graduate student of mine and did some incredibly good field work, and it's really Jay and my work that I'm highlighting here, our lab work. But I'd like to single out especially thanks to Kay Berensmeyer. As you'll see in this talk, I, in the early 1980s, came up with an idea that was sort of theoretical in nature, and I needed a place to test it. And my idea was that we could use stable isotopes in fossil soils to learn something about ecology and paleoecology. Now, I'd known Kay since 1971, when I first went to Kenya. And Kay was in the early 1980s working with the Harvard group in Pakistan. And I don't know exactly where I talked to her, but she said, oh, we have these wonderful sequences in Pakistan for paleosols. Why don't you just come and see what you can find out? And so I just, again, like to especially thank Kay for inviting me, and then the Harvard group, especially Dave Pilbeam, who is the purse string holder, allowing me to bring along Jay Quaid, who is a fabulous field geologist. So that's my acknowledgements. I'm going to talk about isotopes today. So I'm going to give a little primer on isotopes. And of course, you all remember what isotopes are because you learned about them when you were in primary school. And the periodic table of the elements, we all know, is defined by the number of protons in particular atoms. If you have 1 proton, you're hydrogen, 2 protons, you're helium, 3, you're lithium, and so on. And the field of light stable isotopes really uses hydrogen, carbon, nitrogen, oxygen, and sulfur. And what we use are the stable isotopes, and I'll talk about that in a second. But if you think about that, those elements are life. Every biological molecule has those elements in it. And if we can figure out the rules of the biology, then we can learn about biological things. And some of these are preserved in the geological record. And so that's my goal here. So isotopes are really just different flavors, as you will, of particular elements. So as an example, carbon has 3 naturally occurring isotopes. And the difference really is that they have different numbers of neutrons. So carbon-12 is the common variety on our planet, and it has about almost 99% carbon-12. There's about 1% carbon-13, and it has an extra neutron. And a neutron has about the same mass as a proton. And we use that to separate them and measure them. The one isotope that almost everybody who's heard of isotopes has heard about is carbon-14, and that's a radioactive isotope. And it's hardly present on Earth naturally. There's only about 1 in 10 to the 12th atoms, carbon atoms, are carbon-14. And I'm not going to talk about carbon-14, but it is an isotope, and it is the one isotope that most people have actually heard of. So my story today will be using the tiny differences, the naturally differences between carbon-13 and carbon-12 on our planet. My terminology, so we measure the ratio of carbon-13 to 12, and we have a special terminology, which we call a per mil notation. So that's like percent with an extra zero added to it in that middle line there. And so we measure the ratio in whatever we're interested in, and we compare it to some standard. And every laboratory in the world uses the same standard. And, basically both the stuff we're interested in the standard is about 99% carbon-12, the abundant isotope. So we're looking at two tiny differences. And so we just compare these to 1. So if the number is positive, there's more carbon-13 in our compound of interest compared to the standard. If it's negative, there's less carbon-13, and the more negative, the more less. So on Earth, carbon-13 ranges from about 1.04 to about 1.14%. And this delta, what we call this delta per mil notation, that's minus 65 to plus 25 per mil. Now we can easily measure to 0.1 per mil. So we have a big playing field, as it were. And so that's my story today, is going to be entirely about carbon-13 and what we can do with it in the Siwaliks. So this was the paper I alluded to in 1984. I published this theoretical paper, and we did a little bit of field work on modern soils. And what we showed is that the oxygen isotopes recorded something about water. That was interesting. But the carbon isotopes recorded something about 2 different photosynthetic pathways. And that's going to be the subject of my talk. And so it was in about 85 that Kay invited Jay and I to come over. And so in 1986, we went over to look at the soils in the Siwaliks. Now, we're interested in the Paleosols, and to this day, the best place I have ever seen Paleosols developed is in the Siwalik sediments. They are simply fantastic. The sedimentation rate is high, and that means that we can have soils that are essentially quenched. They're buried by enough sediment that they're removed out of the soil accumulation zone. There's some really key features that are really important. And diagram A is a modern soil, and the top of the soil is at about the top of the A there, and you can see there's a bunch of white blobby nodules starting about 1 hammer thickness below the soil. So that's the leached zone. That's the leached zone of the soil. And then there's these white carbonate nodules. And carbonate, of course, is calcium carbonate, so it has the carbon isotopes in it. Now, B is a picture of Jay Quaid standing, pointing to the top of a paleosol. The pickaxe there is the very top of the paleosol, and above it is this layers of fluvial sediments that have essentially quenched the soil. So we have this beautiful Paleosol. And then C and D are just a couple of other examples. C shows a beautiful A horizon with organic matter preserved, and then in D, the actual top of the soil is well above the pickaxe. So there's a leach zone of probably almost a meter, and then all that white carbonate. So what's important is that the B horizon is leached of the original any original calcium carbonate. And it turns out the Siwaliks are fantastically useful because the original sediments are about 10% carbonate. So they form this pedogenic carbonate very quickly in the B horizon. Again, the Siwaliks are useful. We have this complete bioturbation in the soil, destroys the bedding features so we can recognize the soil. Another important feature is that there's this approximately 20 centimeters of leach zone. We hardly ever in any modern soil can find carbonate in the top 20 centimeters. And this is important because the theoretical model that I developed showed that there was a big isotope gradient in the upper 20 centimeters of soils, but it turns out carbonate doesn't form there. And so any carbonate that we have can record the information about the carbon isotope value of the soil CO2. So we get this faithful preservation of soil CO2, but as calcium carbonate, and it lasts for millions of years. So this was our first paper that we published in 1989, showing this incredible carbon isotope shift. So I don't know if you can see my cursor here, but we go from these values of minus 10 per mil for the bottom of the sequence in the Chinji-Nagri formations. Then we have this sudden rise in the carbon isotope values to about plus 2 per mil. So we go from minus 10 to plus 2 per mil. So this is a major isotope change, as it turns out, from two completely different photosynthetic pathways. To this day, this is still the best sequence that we've been able to find, this complete transition. I've worked in other parts of the world, and the transition usually is much more gradual. So this is just a fantastic demonstration. So what I want to talk about today are some of the implications for paleontology and for paleoecology. And in particular, I think what would be useful to talk about is really what is the fundamental difference between these two photosynthetic pathways. So here's the difference in isotope space between the C3 and the C4 plants. And you can see those are not the same values as the calcium carbonate. There's about a 15 per mil enrichment between the CO2 produced by the plants and that it actually is preserved as calcium carbonate. And this diagram shows a survey that I did of about 800 plants, C3 plants, which have these values of minus 25 to minus 30 or so per mil, and the C4 plants. It turns out that all trees and almost all shrubs and most herbaceous plants use the C3 pathway. And it's only the tropical grasses and sedges that use the C4 pathway. So here's two pictures, a C3 forest and a C4 grassland. This has huge implications for paleontology because some animals eat grass, some animals eat the leaves of trees. It's also really important. Almost all of the toxins that are produced by plants are C3 plants. C4 plants don't have very rarely actually have toxins that they produce themselves. And so clearly there's a difference in digestive strategies to get at the plants. And so clearly it's important for paleontology because it's a dietary choice. What are you going to have for dinner tonight? Are you going to eat the C3 plants or are you going to eat the C4 plants? Well, let's just say at 10 million years ago, there was only C3 plants to eat. So all the animals would be eating C3 plants. But once this new food shows up that's not loaded with toxins, but is harder to digest, what is the strategy of the animals going to be? And so it's a great opportunity to test paleontological hypotheses. Paleontologists are always coming up with ideas of things. Well, here's an animal whose tooth morphology changed, and it's because it started eating grasses, is the story. Well, we can test that in a completely independent fashion that's independent of the morphology. So our notion is that, well, if an animal eats C4 plants, then its tissues should show up. So the hair, the meat, and the teeth should all record that in the diet. Likewise with C3 plants, an animal that eats C3 plants, it should show up in the hair, the hoofs, the meat, the teeth. So it's been very useful for modern ecology. We've actually applied this and learned all sorts of unexpected things in modern ecology, but it's incredibly useful in the paleontological record because it's preserved in teeth, in fossils. But when we first started this work, that was not widely believed. So let me just summarize this difference between C3 and C4 plants. C3 plants are the early photosynthetic pathway on Earth, based on compounds produced by photosynthetic organisms. We can show, Kate Freeman at Penn State has shown that there were C3 plants back 3 billion years ago. And at that time, atmospheric CO2 was actually about 5 or 10% CO2. Today, CO2 is only 400 parts per million. It's actually a trace gas. Oxygen is the abundant gas today. Back when C3 plants formed, oxygen was a trace gas. So CO2 and oxygen are reversed in their abundances. Almost all dicots use the C3 pathway. Cool season grasses use it, so at really high altitudes. And this is puzzling. Why does cool season grasses use it? And I'll come to that in a bit. So if you go to the grocery store, you can buy C3 plants. All the vegetables are C3 plants. All the fruits are C3 plants. All the beans, all the lentils are C3 plants. And then there's a few cool season grasses, the wheat, so the bread is a C3 plant, barley and rye. And if you buy meat, it'll depend on what the animal ate. So C4 plants evolved in low atmospheric CO2 conditions. And I'm going to spend quite a bit of time today talking about why that is. It's very rare in dicots, and we find it's the tropical grasses. And what grasses are those? For the foods that humans eat, it tends to be maize, sorghum, sugarcane, millet, teff, fonio. Teff and fonio are two African grasses really important in some of the local diets. And then again, animals, they will tend to show up, they will tend to look like the foods they eat. So interestingly, sheep tend to eat the grasses and goats tend to eat the dicots. So if we analyze the isotope values of coexisting sheep and goats, often there's almost no overlap in their isotope values, even though they're grazing in the same place. Cows will go for the grasses. Well, we wanted to use this in fossil teeth, but at the time it wasn't believed that fossil teeth would preserve isotopes in diets. And so again, the Siwaliks were actually, this was a key paper, Jay Quaid's the first author, that demonstrated that C4, that the diet shift was actually preserved in enamel. And the reason it wasn't believed is because some work had been done on bone, and it was shown that bone didn't preserve in the geological record because it's reset during diagenesis. And so tooth enamel was swept into the same trash can as the bone. And so this was a key paper, and the Siwaliks then were very key in demonstrating that, in fact, fossil diets could be preserved on geological timescales. And so this was incredibly important for paleontology because then we had an independent way to look at browsing and grazing in fossil mammals. And that's a project, that's something I've been following on now for the last nearly 30 years of trying to look at modern ecosystems and at fossil ecosystems and understand diets. So what does this mean for paleontology? Well, this means we can test some of these hypotheses. So our idea was, wow, we've got something here. The classic story in paleontology of changing from browsing to grazing was North American horses. And this interpretation has been around since the late 1800s, was that equid hypsodonty was related to grazing. Teeth got tall, and therefore they were grazing. This transition, as you can see in this illustration by Catherine Stromberg, but the idea goes back actually into the 1800s. Tall teeth means grazing. And again, talking to the paleontologists and paleoecologists, say, well, what kind of grasses? Well, the same as today, which would mean C4 grasses. So we went out to test this, and to our great surprise, it wasn't at all related to hypsodonty. Here's North America. We see the isotope transition is very abrupt at about 7 million years ago. So it's not related to hypsodonty. And the other really interesting thing was that in Pakistan, so we were concentrating here on, it's mostly equids, but we have some Proboscidean data. We see that in Pakistan, it's at 8 million years ago. In North America, it's at 7 million years ago. In East Africa, it was at 8 or 9 million years ago. So, and in South America, 8 million years ago. So it's a very similar time, not quite identical. It appears that the tropic, the absolute, the lowest latitudes went first, followed by intermediate latitudes, and then a little bit higher latitudes. You get to really high latitudes, like in North America, it happens way, way later. And in Western Europe, it never happens at all. Well, this has to do with the temperature relationship. as to some of the delay, but this abrupt nature where it shows up in Eastern Asia, Pakistan, East Africa, North America, 3 widely separated continents within about 1 million years of each other, suggested to us it's a global phenomenon. And in talking to my colleague Jim Elringer, he said, well, it's CO2. I mean, we know that C4 plants are adapted to low atmospheric CO2. And so that's the topic that I want to talk about, because this is really a rather important and interesting topic. And what, there's an idea, well, it goes aridity, but we've had arid environments on planet Earth for the whole time. I mean, we have lots of arid environments in the Eocene and in the Cretaceous, and C4 plants didn't show up in those arid environments. So it didn't seem to be an aridity thing, but we thought, well, it could be atmospheric CO2 falling below a critical threshold. So I want to talk about how that works. First, a quick summary now. We've basically almost no evidence of C4 plants before 10 million years ago anywhere. So today, the net primary productivity in the terrestrial ecosystems is about 25%. But below 30 or so degrees latitude, it's actually about 50%. So in the tropics and subtropics, C4 plants make up 50% of net primary productivity. This is one of the biggest and most abrupt ecological changes in Earth history. It is essentially the same as land plants, the same as angiosperms. This is an incredibly big ecological shift, but we have a really hard time seeing it because C4 plants as fossils are hardly ever preserved. We're left with this shadow isotope way of seeing it. First, now I want to talk about the difference between the C3 and the delta C13 values. Why are these values different? It has to do with the anatomy and the physiology of the plants. So on the left, we see the Rubisco cycle. CO2 goes into a C3 plant. It goes into it's carboxylated by Rubisco. We end up with a bunch of useful products, sugars and starches that are made, and oxygen is expelled from the plant. The green places on the left is where photosynthesis occurs. It occurs everywhere in a C3 plant. C4 plants do something different, and it's called C4 because the very first product is a C4 acid. So CO2 is carboxylated by PEP. It's made into a C4 acid. Oh, somewhere else on the inside, in this bundle sheath cell, which is right in here, the CO2 is now expelled, and the Rubisco sees it and takes it up, goes into the Calvin cycle, and we get net photosynthesis. So photosynthesis takes place only inside these bundled sheath cells. This is a very different situation. So what happens with, well, let's back up a second here. I'll show this again later, but this where Rubisco is seeing the CO2, it's essentially seeing atmospheric CO2. What happens in the C4 plant is CO2 is concentrated. This is a CO2 concentration mechanism. So CO2 inside the plant is 2,000 ppm. Here, it's like 300 to 400 ppm. So Rubisco here is seeing the true atmospheric CO2 level. Here, Rubisco does not see CO2 levels. In isotopes, we call this an open system versus a closed system. In the C3 plants, the Rubisco enzyme is allowed to discriminate. It's allowed to choose the isotopes it wants. It chooses the light carbon-12 isotope and leaves behind the carbon-13 isotope. So its delta C13 value is very negative. In C4 plants, it's a closed system. It has to use all of the CO2 in the system. This is kind of like when you feed your 4-year-old peas and carrots. And this is the top of the slide, shows what's presented. In an open system, you can choose. And in the left side, we see this choice has concentrated the carrots relative to the peas. And so the ratio of carrot to pea is different than in the starting material. In the closed system, we have to use all of the, we're not allowed to choose, so there's no discrimination. So the ratio is identical to the source. And that, therein lies the difference between the C3 and the C4 plants. So I want to follow up on this further, and this idea of C4 photosynthesis and how it's adapted to low atmospheric CO2. So you've all done experiments in chemistry, and one of the things that is a general rule in chemistry is that all chemical reactions increase at higher temperatures. Now in plants, we have two different competing reactions. We have photosynthesis, where Rubisco absorbs CO2 and gives off oxygen. That's a good reaction. That's the positive reaction. The bad reaction is photorespiration. And in photorespiration, RUBP absorbs oxygen and gives off CO2. Now, it turns out that both of these reactions are functions of temperature, but the oxygen, the bad reaction, has a higher slope. So if we think what happens in oxygen to CO2 ratio in C3 plants, oxygen is 210 parts per million in the atmosphere. CO2 is only about. Somebody else is giving a lecture.
Mukhtar, you need to mute yourself.
So CO2 inside of a C3 plant is only about 250 ppm. So the ratio of oxygen to CO2 is about 800. So in spite of there being way more oxygen, plants still prefer the CO2. But if this bad reaction goes fast enough, it can overcome the good reaction. Now that doesn't happen at all in C3 plants or C4 plants because CO2 is concentrated on the inside of the bundle sheath cells. So the oxygen to CO2 ratio is only something like 100, 210,000 ppm to 2,000 ppm. And so this bad reaction doesn't occur at all. So in high temperatures where the bad reaction takes place in C3 plants, eventually C4 plants can win out. So this is basically a case where the bundled sheath cells, because of this concentration mechanism, have overcome this deficit. So actually C4 plant, the C4 photosynthetic pathway, is an adaptation to the low atmospheric CO2 that we find at the end of the tertiary geological period. I'd just like to look at what happens during a typical day just to think about temperature ranges and how this would affect things. This is 3 sites in Africa that we monitored, and we were able to determine the soil ground surface temperature on the average day over a 3-month period. So in East Africa, day comes up at dawn is at 6, nighttime is at 6 p.m. In the grasslands, we reach ground surface temperatures of up to 60 degrees Celsius. What that means is that this time of the day is just way, way too hot for any C3 plants to exist. So this is a time in which the C4 plants in the grassland is favored. In the riparian forest, it doesn't get that hot, and so we can actually, because of the shading of the leaves, we can actually get C3 photosynthesis still going on in riparian forest, but on the grassland, it's simply way, way too hot. There's another way of thinking about it. We could look at the oxygen to CO2 ratio. And so this is what the actual plant is seeing. In a C3 plant, external CO2 at 350 ppm. The internal CO2 where the photosynthesis takes place actually is about 100 ppm lower. So in C3 plants, we're down in this range. But again, we've got 210 ppm oxygen to go with. So in the atmosphere, as CO2 in the global atmosphere increases, we're reducing this this gradient in C3 plants. So we can see at really low glacial conditions, where CO2 is 180 ppm, we're at a very huge disadvantage for C3 photosynthesis. So C3 photosynthesis is a very hard time in this region. When eventually we get out to this area where there's no net advantage of C4 photosynthesis, C3 plants can always make it. So let's look now at how this fits in the geological history of CO2. So this is a famous picture by Lysick and Ramo showing the benthic stack of oxygen isotopes. And we see a couple of important things. There's some really big changes in frequency of the isotope transition for the last almost million years. We have this 100,000 year cycle. Then we get back to this 40,000 year cycle. We get back into the Pliocene. We're in another climate regime. In the last 20 years, various people have been developing CO2 barometers. This is a barometer. Of course, we have the ice core records, but Tripathi et al. Has developed a boron isotope barometer. This shows that the 180 to 280 ppm record goes back at least into the middle Pleistocene, certainly 800,000 years, maybe even 1 million years ago. In the early Pleistocene, we're in a slightly different, we're in a different rim. We're not getting these really, really low values anymore. There was a nice paper just published earlier this year by James Ray and a whole bunch of other colleagues in annual reviews, in which they've reconstructed atmospheric CO2 for the last 60 million years. Here's the last million or so years, between 180 and 280 ppm. From about 1 million then to back about, curiously here, about 8 million years ago, we cross over this period of 400 ppm. So before 8 million years ago, we're always at above about 400 ppm CO2. This is really interesting because 8 million years ago was the time when we switched from, we had this rapid C4 expansion. So this really lowest period where CO2 fell below this critical threshold, whatever it is, 400, 500 ppm CO2, is right in this period where we see the C4 expansion. So let's go back to this figure that looks at the CO2 to oxygen ratio. Well, here's the glacial interglacial transitions between 180 and 280 ppm. We're in this part of the curve. Okay. Then between about 700,000, maybe a million years ago, we're in this interval between 280 or maybe 250 ppm to 400 ppm. And at greater than 8 or 10 million years ago, we're in this period where C3 plants, or rather C4 plants, have absolutely no physiological advantage towards photosynthesis. So I'd just like to summarize. There's a couple of key points here. The C3-C4 transition is really important in variable temperatures across the landscape. Another very important thing is the oxygen to CO2 ratio in the atmosphere. Above about 400 or 500 ppm, there's no reason for C4 photosynthesis physiologically. And of course, in the context of what humans are doing to atmospheric CO2, that's an interesting situation. So atmosphere, or so C4 plants, the C4 pathway evolved in response to low atmospheric CO2 and probably the starvation of C3 plants. And then I'm going to leave you with the question of, are there thresholds? Is 400 or 500 ppm a threshold? I'll just mention 3 other interesting things to think about. Each of these is a 1-point slide. There are really some wonderful opportunities to use the isotopes to look at the C3 and C4 shift. In paleontology, test these long-held assumptions about diet and morphology. Equids, I already showed you an example of equids. Other animals too. Elephants, giraffes, suids, primates, all of those have big morphological changes in tooth or other anatomy that people have attributed to diet change. Another thing is, well, let's just look at one of those. Here's elephants. Here's a case where the diet shift in elephants happens before the hypsodonty shift. So the diet switch in elephants happened before the change in hypsodonty. I showed you the thing in equids where at least the C4 plants showed up after the equids had already become hypsodont. Giraffes, the Thure is an example of an animal that becomes hypsodont and then begins to eat C4 plants. Suids in Africa, primates, all the primate groups tried C4 diets. And so again, this is a good independent test of some paleontological observations. Here's another interesting thing, is there's been a recent shift away from C4 plants in both elephants and primates. And this is a paper we published in 1999. So this problem has been kicking around for 20 years, and nobody's been able to explain why elephants have gone back to eating C3 plants. They were happily eating C4 plants for millions of years, and in Africa, 100,000 years, they've switched back to C3 plants. Why? Primates have done the same thing. All the major primate groups tried C4 diets, and they've all switched away from it, except for humans. Then one other last one. This is a really interesting one. There's a big marine isotope shift in carbon isotopes. And look when it happens. 7 million years ago. It turns out that if you convert 25% of the global terrestrial biomass from C3 to C4 plants, you get at least half of this isotope shift. Right now, the marine people are proposing that this is totally something that happens in the marine realm, but I think it could be in the terrestrial realm. So in summary, the Siwaliks provided a really key kickoff to understanding this global expansion of C4 biomass. It's really interesting looking at the timing and how this happens in different places. This is a clear, it's definitely related to the history of atmospheric CO2 on our planet. And lastly, I think asking various questions in paleontology, you can usually use isotopes to provide some really useful independent tests of paleontological hypotheses. So with that, I'm going to close and hope you've enjoyed this talk, and I'm answer any questions.
Yeah, thank you very much, Thure, for a wonderful review of this tool and these systems. And I think it's spectacular for us as an audience to hear your account of the history of understanding that you've watched and witnessed with together with your students, and a wonderful presentation. I'll just start off by posing the question about this the global diachronous nature, the different timing, and I hear your primary suggestion is that it's because of latitudinal differences and average temperature differences. And is that something that you could have you sort of made maps of that show sort of a sweeping change that
it's not really, there's very few sequences that where you can actually liken the Potware Plateau, where in one location with a limited latitude you can look at that. And in fact, if you actually go east along the Siwalik, along the Himalayan front, the timing is not even synchronous along the Himalayan front. And in fact, you get into Thailand and there's no isotope change at all. But there's like in North America, we find a really good section that covers 2 million years here, and we have to go to Texas to find a different 2 million years, and California to get a different million years. And there's very few places where we can actually look at it as you have suggested would be very nice. So we kind of are almost restricted to continent by continent.
Yeah, I think in her presentation, Lisa Taux a couple of weeks ago also indicated that there was a diachronous pattern between Pakistan and I think Nepal.
Yeah,
and that's startling given that the distances are not that great, and actually the obviously the latitude is not very different at all, and so that's a surprising
yeah.
result.
Yeah, and actually in Australia,
I'm happy to ask the follow. Yeah. Oh, go ahead, Thure.
I was going to say in Australia, the C4 plants almost don't seem to show up until about 4 million years ago at all. And Alison Karp, who has done some really beautiful work on showing that fire is also very important in showing up at about the same time as the C3 to C4 shift. It looks like the fire regime in Australia may be different, and so that may also be part of the story.
Mm.
I was just going to ask something about those modulating factors and the role that precipitation might, whether or not there's any role that precipitation might play, since precipitation and temperature, at least locally, can interact. And I'm thinking back to the difference between Pakistan and Nepal. Are there other modulating?
I seem to have lost you. I don't know if anybody else is maybe I'm lost too.
No, you're here. She seems to have just dropped out briefly,
Yeah.
but she did
But
post.
yeah, her question was there other modulating factors, I think. One of the really important things is that the grasses are very well adapted to fire because their reproductive, many of their reproductive strategies are underground. And so they have a good protection for fire. Alison Karp, in her PhD thesis just completed just a year ago, has been working on these fire biomarkers and also has been able to develop some aspects about the temperatures of fires. Anyway, she's developed these markers, and we're seeing that they seem to change at about the same time as the C3, C4 transition. It's something that would really work together very well. And obviously, seasonality can play in there very nicely as well. One of the things that we do know is that it can't only be aridity. There's always been arid places on planet Earth. It can't only be seasonality. There's always been high seasonality in some parts of the planet. And likewise, we also know there's also been some fire, but fire frequency does seem to change at about this time.
So sorry, I just got bumped out somehow and rejoined. But I was a question, and you may have already addressed the question that I was trying to pose, but I'm wondering what about areas that are so arid that we would call them deserts today? Why do we not see many C4 plants in those places? Because the temperatures would be high, precipitation would be low, but there seems to be apparently some threshold that favors C3 plants once again.
There is, and those particular C3 plants generally have developed an independent strategy, often for cooling, that basically can help them out. And I think it is an important thing about aridity, because actually C4 plants, if it's really arid, they don't always do that great. They're in the tropics and subtropics, their favorite rainfall, window is about 500 to about 1,200 millimeters of rain a year. And 1,200 millimeters of rain a year doesn't fit anybody's definition of aridity, of being arid.
Right.
So Chicago gets a meter of rain a year. And it was a native grassland until 200 years ago. So I don't know if if you could think that's a sidestepping your question, Catherine, but a lot of those desert, you are right that if it gets too arid, the grasses themselves don't do well. But in talking to my ecological colleagues, there's other strategies for cooling plants. like precipitating salts on the leaves and developing really thick, waxy leaves to prevent water loss.
Terry, let me just ask another question related to the thought of thresholds. And as we now are entering into a situation where the PCO2 is climbing back up again to what might have been a threshold that you indicated, do you see are there going to be global influences on flora, on vegetation as a function of the changing atmospheric composition?
I think there will be, but other things are going to be so huge that teasing that out is going to be very difficult. I mean, first of all, in most places on Earth, we're living on an engineered planet. We are engineering the planet and making choices that the natural ecosystems don't make. And the temperature rise is so fast that it might be larger than the CO2, than any CO2 effect. So I have a feeling that the answer is yes, but it's going to be a second order effect because changes in rainfall, changes in human activity, and changes in temperature are going to be probably larger in this next 100 years than the CO2 effect.
Terry, I'll ask a question since others aren't coming up yet about the value of studying modern ecosystems from the standpoint of the stable isotopes of carbon. I know you've participated in some really interesting, what I might call faunal surveys from sites in, for example, I know at least of your studies in East Africa, and you and I have at least talked about the idea of doing a similar survey in a monsoonal grassland in South Asia somewhere in order to look at the distribution of the carbon isotopes across the vegetation and the mammals. And I thought it would be interesting for the audience to hear about your experience doing that kind of research and what you think the future holds for those opportunities in Pakistan and India.
Absolutely fantastic opportunities. You know we've found a lot out about. I mean, so for instance, in Africa, we've been able to reconstruct the annual diet changes in animals without even seeing the animals, except twice a year when we collect hair samples, because the hair can so the hair is recording stuff whether you're watching the animal or not. And it's provided a lot of other new insights into animals, because often, behaviors aren't as when we are watching the animals, it's often anomalous that you can actually see the animal. And so I think there's really great opportunities. The tricky part is going to be actually the analysis. I think in India, it's really difficult to export actually modern materials, but there are plenty of labs in India that can do the work. I'm not aware of any labs in Pakistan that are doing that right now, but there's just absolutely fantastic opportunities to work on modern ecology, and then that in turn gives insight into ancient ecology. I'd say for students or people just starting out now, there's some really fantastic opportunities.
So let me, I'll just pop in again, and it might be we're sort of drawing to a close here, but Thure, in your studies in North America, I can appreciate that the record is not as good as it is. And I mean, one of the wonderful things about the Siwaliks is you've got this marvelous continuous record. But if we go to, for example, the Bighorn Basin, or I'm trying to think if there are other places where you might have something comparable to what we have in Pakistan, and whether those are areas where people are continuing to do research along these lines.
There's lots. Let me just go back to, again, and this might be a lesson for some of the younger people. Once we worked out this thing on the Paleosols, I found it incredibly frustrating because there was a lot of people who were not paying attention to the rules and the guidelines that we had set out. And so people were calling everything Paleosols. And basically, if they didn't know what it was, they'd call it a Paleosol. And I remember I visited a place in England where a guy was showing me an oolitic limestone, and he was calling it a Paleosol and analyzing it for carbon isotopes, and saying there were C4 plants back in the Ordovician because of the carbon isotope value. And I said, well, why don't you call this a limestone? An oolitic limestone. There were actually oolites that you could see. He said, well, because it's a Paleosol. And so that's one of the reasons I decided to work on mammals, was because it was so frustrating with other people not following the guidelines that we had set out for Paleosols, and everything was a Paleosol. And I figured, well, at least with a horse tooth, I can tell, even I can tell the difference between a horse tooth and an elephant tooth. So
Mukhtar,
I see Kay have her hand up. Yeah.
I do. Thure, that was great. Very worth staying up late to hear you again. And thank you for all your kind words. It's wonderful how much has happened since the 80s, when we first thought the Siwaliks was a great place to look at Paleosols. But the Paleosols also, some of them interspersed between the ones with a lot of pedogenic carbonate, don't have pedogenic carbonate. And yet they're very extensive laterally, they're thick. So we're thinking they must represent some other climate state. And I wonder if you might want to comment about soils that don't have carbonate and what they might mean.
A couple of things. One is that it'd be really nice to know, in those sequences, was there originally carbonate deposited as a detrital phase? Because one of the things to produce calcium carbonate, you have to weather something that produces calcium. And if you actually have 10 or 15% calcium carbonate, that provides calcium. And you leach it, but then you precipitate it somewhere else. So on those sequences, it'd be nice to look at the intervening bedded material, and does that have carbonate in it? That's just another important thing, because you have to weather something to produce calcium for the carbonate. And then, of course, really high rainfall regimes, you don't get calcium carbonate. And if you don't have enough time, you don't get calcium carbonate. So it could be a combination of those, of all of those. Is that close enough an answer?
Yeah, thanks.
Yeah, Samina?
I think it's all the same parent material, so I suspect that it's a changing rainfall.
Yeah, but so we analyzed the parent material in the Poppy Hills and in the all of the groups that we did our first survey on, and the original parent material was typically 7 to 15% calcium carbonate, which really was nice. If it would have been down at 1 or 2 or 0%, which could be weathering in the uplands, higher rainfall in the uplands may do the leaching. So it wouldn't have to be a change in the source region. It could be a change in the climate of the source region, but they'd be really easy to test.
Mm-hmm. More to do.
Yeah. Samina Siddiqui?
Good morning and good evening. Can you hear me?
Yes.
Yes,
Professor,
yes.
it's a very interesting talk, and I read a lot of your papers and including as a reference. But I just want to clear some of my doubt that I am thinking of looking at the microbial aspect of these C4 and C3 vegetation, which are preserved stratigraphically in different geological period. Do you think it can be related to the Paleosols and paleoclimatic or paleo-environmental conditions can be established with the types of microbes I'm going to isolate from C3 and C4 isotopic analysis samples? And can I do it from the surface, or can I do it from deeper, horizontally deeper inside the rock, so that the conditions will be more preserved than exposed to the environment?
Yeah, I really don't know. There's, the preservation of organic material in these is low, but I think if you get into the right localities, certainly the people working on biomarkers have been able to find organic material for the biomarker works all the way back to certainly 12 million years ago. My guess is that you're going to really have to be worried about surface contamination. So if you can get really deep within an outcrop, like maybe in road cuts, that'd be the best place to try to get the material it sounds like you're trying to get. Does that have help at all?
Okay. Okay, thanks.
And or some possibly some shallow drill cores. I think I'm always so worried about the upper 1 or 2 meters of weathering, because I know roots and stuff. So I'm just always so worried about organic contamination in the near surface stuff. But often road cuts provide somebody else did the digging for you.
Mm-hmm. Okay, thanks.
All right. Well, thank you very much, Thure, for a wonderful presentation, and we certainly appreciate your contributions. And good to have a discussion and some good questions here. And I'd like to invite everybody to join us a week from tonight or this morning for the talk by Shahid Iqbal on the provenance of sediments in the Peshawar Basin. So thank you all for your participation in the seminar, and we'll look forward to seeing you next week. Thank you.
Thank you.
Thank you.
Bye-bye.
Yeah. Thank you very much, Thure.
Bye-bye.
And thank you guys on the East Coast of the US for being up in the middle of the night.